Good day, we'll be talking about the dark phase of photosynthesis, also known as the Calvin cycle or the light independent phase. Now, this phase occurs in the stroma of the chloroplast. The stroma is the fluid-like substance found in the chloroplast. The same like in a cell, we have a jelly-like substance called cytoplasm, or in the mitochondrion we have the matrix. Now in the chloroplast we have the stroma. So carbon dioxide is the source of carbon and oxygen from which glucose will be built. Remember, in the chemical formula for glucose we have 6 carbons, 12 hydrogens and 6 oxygens. Now where we get this from, these compounds, or not these compounds, these elements, is from carbon dioxide. Carbon dioxide is absorbed into the leaves from the atmosphere. Remember, the requirements for photosynthesis we said was carbon dioxide. And hydrogen atoms, they are supplied by the NADPH from the light phase. Remember the coenzyme NADPH. I see that it will be carried on into the next phase. So that is where we get our carbon, our hydrogen and our oxygen. And don't forget also ATP. ATP is also carried from the previous step into the Calvin cycle, but I'll explain further on. So we are now in the Calvin cycle. A C5 compound called what was it called? Oh, it was called ribulose biphosphate. You don't have to know that name, but that's just the name of the C5 compound, ribulose biphosphate. So it's a C5 compound that has phosphorus, okay? It has phosphorus on both sides, so it has one attached to the first carbon chain. So over here you would have one phosphorus attached here to the first carbon chain, and you have another attached to the fifth one. So it's going to be 1.5 ribulose biphosphate. 1.5 meaning attached to the first and fifth carbon chain. So this is already in the plant cell, in the stroma. And then the carbon dioxide, which was absorbed from the atmosphere, comes along and it bonds to the C5 compound and it forms a C6 compound. If you look in this bondage, or this chain of bonds, I know you can't see the oxygen, but just know it's there. The main focus is the carbon. That's why you won't see the oxygen and the hydrogen specifically in here in each bond, but the oxygen is there. So remember, the C5 compound binds with carbon dioxide from the atmosphere to give a C6 compound. So this is now, okay, it's an unstable compound, an unstable C6 compound, and then breaks to form two identical C3 molecules known as PGA, phosphoglyceric acid. These are low energy trios. A triose is a molecule that has 3 carbons. So this PGA has 3 carbons in it and it's low in energy. But remember, our main product, we want to get glucose, and glucose is something that has high energy. So this is how we'll get that high energy. So like I explained, here's the RUBP, the ribulose biphosphate. See, it's attached, it's 1, not 1,5, 1,5 ribulose biphosphate. One phosphorus is attached to the first carbon, and another one is attached to the fifth carbon. So don't worry about these big numbers. You see these big numbers here in front? Don't worry about it. I'll explain further on. We have carbon dioxide comes from the air, and it binds and fuses with the C5 compound. C5 meaning it's 5 carbons. It now is a C6 carbon, but it becomes unstable. Oh yes, the enzyme that helps catalyzes the reaction between the ribulose biphosphate and the carbon is called Rubisco. It's an enzyme that helps speed up the reaction between the two. So we left with the C6 compound, but that compound becomes unstable, and it, like we said, it breaks into two identical C3 molecules, and it's known as PGA, phosphoglyceric acid. When you see there, there's the three carbons, there's the one phosphorus, there's the other phosphorus. So it would most likely just break down in the middle, so it's identical. And remember, we want energy rich trios, right? We don't want energy low trios because glucose has a lot of energy in it. So remember the ATP from the previous step, we said it's going to carry on to this step. Now the ATP is here in the organelle, and remember I said there's energy between the bonds of the phosphorus. There's an energy between the bond. So remember, it was originally adenosine diphosphate, but it became adenosine triphosphate, and it was known as an energy carrier because there's energy in the bond. Now that bond between the ADP plus the phosphorus breaks, right? And energy is now released. That energy is now added to the PGA. So you see we increase in the energy, and now the ATP becomes ADP plus P. So it's gonna result in adenosine diphosphate and a phosphorus. That two molecules can be reused again in the organelle for photosynthesis. So they won't disappear or just it will become non-existent. They will still be reused again in the light phase. So we now increase the energy for PGA, but we still have NADPH. Remember, NADPH, it's a coenzyme. The NADP is a enzyme carrying a hydrogen-rich atom. No, not a hydrogen-rich atom, an energy-rich hydrogen atom. And what do we need? We need energy. So how are we going to get that energy from the NADPH to the PGA or the C3 compound? There, the NADP is now going to release the hydrogen. It's going to drop it off. Remember, the NADP is just a carrier, like I showed in that diagram before, the Paki being the NADP and the H where it's been picked up. Now this is where the NADP is going to drop off the H, the hydrogen atom, the energy rich hydrogen atom. Now the hydrogen atom then bonds with the C3 compound, and that also increases the energy levels. Now when energy is gained, right, when a compound gains energy through electrons, it is called reduction. So over here you can see reduction. So here energy was gained from here and gained from there. So that is known as reduction. Reduction is basically when a molecule or chemical reaction and the molecule gains one or two electrons, and hydrogen is basically an electron. So if this NADP loses an electron, it becomes NADP plus. That's why it's NADP plus, but you don't have to worry about that. So remember, ADP, we lost energy. The bond between ADP and the phosphorus was broken. That resulted in ADP, or the bond between ATP. The bond between ADP and P was broken, and now we just left with ADP and P. There's no bond between the two. That is reused in the light phase. The energy was added to the PGA, C3 compound. NADPH comes here. The NADP drops off the hydrogen atom, energy rich, which is basically an electron. That hydrogen atom attaches itself to the PGA, right? The PGA again increases with energy, and now the NADP becomes a plus because it lost an electron. The hydrogen was basically an electron, and the NADP is again reused in the light phase. So you see how we reuse stuff here, and now we're left with two energy rich C3 compounds, right? Remember, there is oxygen and there's more there is more oxygen in here. It's just not showing, and in this diagram it's not showing the hydrogen attached to it, but it's three energy rich C3 compounds. This C3 compound is known as phosphoglyceraldehyde, but they call it glycerol dehyde phosphate, something like that. It's the same thing, just different. They say it different, so that's why they have G3P, but we would have PGAL, phosphoglyceraldehyde. So phosphoglyceraldehyde is two C3 compounds that are energy rich. This compound is then taken, it leaves the cycle to make glucose. Okay, so now we have glucose, but remember, in a cycle it must be continuous and must be never ending. So how does it go again from, how does it go back to a five carbon compound, a C5 compound? Well, this process actually occurs 6 times. So think of it like this. You have, let's say now, you have this over here. Here is yeah, you have 6 PGAs. Remember, a PGA is a 2-3. Okay, wait. You have 6 PGA molecules. A PGA is 2 C3 compounds. So basically, we're having 6 of 6 carbon compounds. Okay. So in total, it will be 36. Right? This process must happen 6 times. It must go 6 times. So if you have 6 times 6 carbons, right, it's going to be 36 in total. Okay. Now, out of the 36, 6 is going to be taken away, removed to make glucose. And you left with 30, right? And in the beginning, we needed 5. We needed a C5 compound, okay? Now just listen, see if we can make sense out of this. I know it's a bit difficult to understand, but you needed 5 in the beginning. This happened how many times? 6 times. 6 times 5, right? 6 times 5, how much is that? That's 30, right? And there you see how it equals now. It equals, because remember, we said the cycle must happen 6 times. And for every time it happens, there is, in each cycle, there's going to be 6 carbons, right? It's 3 C3 compounds, but in total it's 6 carbons for the cycle. But if the cycle happens 6 times, it's going to be 6 times 6 is going to be 36 in total. But for this to equal, they must be equal in the beginning and in the end, right? In the beginning, oh wait, and remember, one is going to be removed. One of the C3 one of the PGAs is going to be removed, and PGA is 2 C3 compounds, so 6 is going to be removed. So at the end of 6 cycles, 30 is going to be removed, right? So in the beginning, we also have to have 30, okay? So in the beginning of each of these 6 cycles, how much did we need? We needed 5. This happened 6 times. 6 times 5 is 30. So we had 30 in the beginning and 30 in the end after we removed one of the PGAs. So I don't know if you can make sense, but just know the first part that I explained, the first part. You don't have to exactly know the regeneration part, how it is continuous, if you could understand what I'm trying to say. So remember, we had an initial of, it was already in the stroma, we had a C5 compound, ribulose biphosphate. It was 1.5 or 1,5 ribulose biphosphate. It was a phosphate attached to the first and the fifth of the carbon chain. Here we have carbon dioxide coming from the atmosphere. The enzyme called Rubisco catalyzes the bond between the two, and that C5 compound becomes a C6 compound. But the C6 compound is unstable. Therefore, it splits evenly. It splits into 2 identical C3 compounds. As you can see here, C3, yes, C3 is a phosphorus, This C3 compound is known as PGA, phosphoglyceric acid. Some people call it glycerate, but no, phosphoglycerate, but it's the same thing as saying phosphoglyceric acid. But this is an energy low triose. A triose is any compound that has 3 carbons in it. So this is energy low. We want it to be high in energy because we need energy to do things. The body, any organism needs energy not to produce, but to undergo functions and do things and do work. So how are we going to get this compound to increase in energy? From the previous phase, the light phase, the ATP that was the end product of the light phase is carried to the stroma. There is energy in the bond between ADP and P. That bond is broken and energy is released. That energy that was released is added to the PGA, right? And then we're left with just ADP plus P. That ADP plus P can be reused again in the light phase, so it goes back to the light phase. Okay. So now we increase the PGA, the energy in the PGA, but we can still increase it even further. The coenzyme called NADPH, nicotinamide adenine diphosphate with the hydrogen, comes now from the granum to the stroma. Remember, everything here is happening in the stroma of the chloroplast. The NADPH is a coenzyme. It is a non-protein molecule with a enzyme. And this coenzyme is high in energy because of the hydrogen. The hydrogen is energy rich. This enzyme drops off the energy rich hydrogen atom, and that energy rich hydrogen atom attaches itself, it fuses or it bonds itself to the PGA. So we now increase the energy. That NADP becomes NADP plus because energy was lost, and the energy that was lost was an electron. That's why it's a plus. If you lose an electron, you become more positive. And electrons carry energy. Okay, that NADP plus is now transports itself back to the granum, and it can be used again in the light phase. This whole process, or not this whole process, but this small process is called reduction. Reduction is when a molecule in a chemical reaction gains more than one electron, or gains an electron or more than one electron. And here it gained an electron. That electron was the hydrogen, and that electron was high in energy. We now have made the PGA into a high energy triose, and This PGA is now known as PGAL, phosphoglyceraldehyde. And that's the Krebs cycle. So here's another diagram. So we had an energy low trials, which was the PGA. See, it was a C3 compound. It had 3 carbons bonded to each other, but there were 2 of those. Remember, we had 6, but they were unstable and they split into 2 identical C3 compounds. Here we have ATP. It undergoes reduction, right? Or not, that doesn't undergo. The bond between the ADP and the P breaks, energy is released and is added to the C3 compound. This whole thing can be used again in the light phase, the ADP plus the P to undergo again photophosphorylation. We have the NADPH coenzyme that came from the light phase into the dark phase. This hydrogen atom here is energy rich. Remember, we want to increase our PGA in energy because it was a low energy trials. We want to make it a high energy trials. The NADP releases it drops off the hydrogen, and the hydrogen attaches itself to the C3 compound. Remember, this thing happens 6 times. This whole process happens 6 times so that they can equal. The NADP, remember the hydrogen was now dropped off. The NADP goes back to the granum and is again in light phase. Going from a low energy trials to a high energy trials is called reduction. Reduction is basically a chemical action where a molecule gains an electron or more. Okay, the hydrogen was the electron, it was energy rich, electrons are energy rich. Okay, PGA became PGAL, phosphoglyceric acid turned into phosphoglyceraldehyde. Just remember this small thing: oil rig, oxidation is lost and reduction is gained. So when you undergo oxidation, energy is lost, right? Energy is removed. Sorry, energy is removed. But when the reduction